information is crucial to almost all aspects of life, and in particular in biology. What is information? We are going to address that today. What is biological information? And you can see here in these pictograms, who can read it, actually? If you look at it carefully, it is information, and it has the same title as my presentation, which you cannot read at this moment, but it is information as a foundation of biology. So information, what is information? You see, you can convey information in several ways already, and we are going into this topic today. What is it? So, the questions to be addressed today is, what is life? Is the DNA, which is the basis of life, or the genome, is it a template, or is it a code? What is information in general? Do we have an idea what information is? And what is physical information, or what is Shannon information? Does the DNA or the genome only contain Shannon information, which is in fact another word of statistical information? The other important question is, do we have a biological information concept? Then we have to look into what do the evolutionary biologists say about information, but also what do our creation scientists say about information? Are there natural laws of information? That's also a good question. And what are the implications for the origin of life, or for life in general, or for biology? Do we have a source of information? Do we know a source of information? I think these are very important questions, and I think most of these questions are not being addressed nowadays in biology, in the universities, in schools. And the question is why? And let's have a look into what I encountered recently on the internet. It is an article, Science News for Students. And it says, we are stardust. Okay, we are stardust. And this is a paper actually, if you read it, you encounter a quote. And this is actually from an astronomer of the Indiana University in Bloomington, Dr. Pilakovsky, and she's studying stars. She's a cosmologist. And she feels a deep connection to those cosmic objects, stars, and maybe that is because she's made of stardust. And so are you. This is the message nowadays to our students. You are stardust. You are just material. You are just molecules. Anyway, then I ask, and then I come up with the question to these sort of people who promote these ideas. What is the question? My computer is also made of stardust, but does it also run on stardust? There's a different question. Are computer programs, software and so forth, are they made of stardust? I think this is an important question. So, let's think about it. But first, in the time that we can think about this question, I will first have a look into what life is. Because as scientists, we have to know what we are studying, isn't it? We have to have definitions. Otherwise, there's nothing to study. If everybody has his own definition, there's really nothing to study, but because we simply talk past each other. So we have to define. And the NASA definition of life is very simple. It says it is a self-sustaining chemical system capable of Darwinian evolution. And it must be self-sustaining because it needs energy to do that. So it needs to produce energy. It has to have a metabolism, as we say in biological terms. It is a chemical system. It is molecules, stardust, if you will. And it needs at least a cell membrane because you need a compartment where all these reactions take place. So if you do not have a compartment, it will all diffuse. So there's nothing to contain. So you need at least a cell membrane for life. And the third thing that they argue for is it must be replicating. Why is it? Why does life need replication? Or why needs evolution, Darwinian evolution, replication? Because you have to change over time. And if you stay the same and you do not replicate, You cannot change over time. As easy as that. Then you stay the same. So replication, a replication system, is necessary for Darwinian evolution. We all know that. Darwinian evolution starts when you have a replicating cell. First, otherwise it cannot start. And this is one of the preconditions. And then I think one of the best questions that they ask here is, what is the driving force that makes this system run? This is the best question. And why is this the best question? Because I worked in the lab with so-called cell-free systems. Cell-free systems. What are cell-free systems? You have just a bunch of enzymes that you need for instance, a certain chemical reaction. And how long does a cell-free system last? How long can you use it? If you have optimal conditions, a few hours, and it's gone. So there must be a continuously supply of these biomolecules to function as a cell, as a living system. This is one of the things that we discovered, and this is really necessary. So not only a metabolism that generates energy for your system, no, you have to have a system that is storing something that you need in the future. So you need an information system. So life is maintained, they say here, by energy input from the universe and entropy export to the universe. So this is another definition. They think that energy, if you put in energy in the system, it's sufficient. I say no, it's not sufficient because there's something else that you also need. So the same question comes up. What is the driving force that makes the system run? So if you look in the internet, and I have a lot of friends that I discuss with, this is one of them. He recently wrote an article on what he defines as life. And you see a very important aspect is information. He says life is a far from equilibrium self-maintaining chemical system. So first of all, it is a chemical system capable of processing, transforming, and accumulating information. So this is an important concept nowadays to understand the living cell. Because without this information, where do all these cells get their proteins from, for instance? Their tools. Because we know all these tools are subject to decay. They oxidize, for instance. So you need a continuously supply of new. So you need a sort of storage where you can put from. And this is information. This is what we call information. So we need an information storage. Also, one of our founders of our association, Wort and Wissen, Siegfried Scherer, he also realizes this. If you think about biology, what does it need? It needs information. And he says, life is a biological information or based on biological information and the transmission of biological information. You have to know where you are in your environment and you have to respond to it. You get information from your environment and you have to respond to it. So you have an input and you have an output, and it should be a logical output. So it reminds us of computers. So we're sort of computers. Cells are a sort of computer. And the interesting part is that we need information for it. So I define life as a replicating system, a replicating information system. A replicating information system is a replicating computer. So this is in fact what life is, a replicating computer. It's an information system. Before we discovered all these information molecules in 1953 with the DNA starting, what did we know about biology? And how did we think about biology? How did it come into existence? Matter and energy. In German, Kraft und Stoff. That was all was needed. This is stemming from the 19th century materialistic age, naturalism. Kraft und Stoff, matter and energy, will produce all the building blocks of life, and the building blocks of life need energy here to build a cell, to build a house. This is in fact what also the NASA concept still is. It's failing something, and what is failing? It is a plan, it is a construction plan. That's what you need, a plan, construction plan. And this is failing in a lot of definitions that we find nowadays on the internet and also in the NASA concept. There is missing something. The information concept is missing. And my question then is, why is it ignored? Why does nobody point out that there is information in it? Because it will give you an origin problem, which we will address later on. So let's look into bioinformation a bit. You can extensively look into it. Where do these ideas come from on bioinformation? But I think the first one who realized it was not even a biologist. It was a physicist. It was Schrödinger. And I think we all know Schrödinger from his wave functions and so on. And from his cat probably, Schrödinger's cat. But the thing is, he was the first to realize that there is information in the genome, in life. He called it a code script. He says life is built on genes. Genes, the term genes was already coined in those days. And he says genes contain code script that determines the entire package of the individual's future development and of its functioning in the mature state. The whole trajectory of a living organism is information-based, code script-based. And he also predicted that the unit of heredity is an aperiodic crystal, most likely located in the chromosomes. So he was a visionary person because we didn't know what was in the chromosome. Only a year later, we found out by experiments that the DNA was the carrier of the information. He predicted this. And why did he predict it? Why did he predict about an aperiodic crystal, which in fact the DNA is? It's very aperiodic. It's not repeating, because crystals do not repeat. They can be predicted. The next structure can be predicted. It is ordered and it is organized, but it is not containing information. Ordered things do not contain information. This was his vision, his insight. So there is no information in a crystal, in what is it, kitchen salt, for instance. It's very nice to look at. It's very nicely ordered, but there's no information in it. They are information poor. Ordered structures are information poor. Organized structures are not information poor. We'll see that later. So, 10 years later, we discovered the DNA molecule, Watson and Crick. We all know Watson and Crick. They received the Nobel Prize a few years later. And they were very insightful because together with this Rosalind Franklin, who never got the Nobel Prize because she died before, but she actually was one of the 3 people who led to the discovery. But she did the X-ray fraction. So they could deduce from the structure that she found the structure of the DNA. So she made a very, very important contribution to the discovery of this beautiful spiral that we all know of. So she died of cancer. Maybe it had to do with this X-ray exposure that she was all day working with. And in 1953, it was Crick. I think Crick was the real brain behind this whole project, because he realized it is a code. The DNA is a code. He writes, there is nothing about the chemical properties or shapes of the bases, that is the DNA letters, to ensure that only one amino acid would fit into or attach to the cavities created by the bases, so present in your DNA. You see minor grooves and you see major groove. This is a repetition. So he also understood these repetitions cannot bring forth information. So if it is an information system, it must be in the order of these letters, this A, the C, the G, and the T. And he understood it as a code. And why do we know it? Because he wrote it in a letter to his son. What did he write? He says, now we believe that the DNA is a code. That is, the order of the bases, that is the letters of the DNA, the A, the C, the G, and the T, makes one gene different from another gene. It is very chaotic, if you look at it, but it is a code. So it has a lot of information in it. And he says it is like a book. The 2 different pages of a book look very different, very chaotic, but we translate it in our minds and we understand it. There's information in it. It is code. It has to be decoded. So we are dealing with codes. The DNA is a coded molecule. Quick insights. Here, I sum them up for you. The DNA cannot be explained as a template. And the specific arrangement of the nucleotides, these letters, and he said possible triplets, and he had his reasons for that, which I will not discuss today because it's going too deep. But possible 3 letters specify 1 amino acid. And he says the translation of the DNA sequence into a protein requires the existence of a code, a means of translating information from one domain to another chemical domain. So we need an interpreter. This is also what he realized. So he said there must be at least 20 specific adapter molecules which translate the nucleotide code into proteins. And so there must also be existing 20 specific enzymes to connect the amino acids to the corresponding adapter molecules. So you have a DNA which has to be translated in a protein. So you need to do that in chemical steps. So this is what he predicted. And why did he know this? How did he know this all? Because he knew, or he thought, it's a code. So you have to decode it. And these are the minimal requirements for decoding the DNA system into proteins. Because we know there are 4 letters in the DNA, and there are 20 amino acids in the proteins. So this must be translated. So code, it's code. The code is a means of translating information from one domain into another domain. So what is the one domain? I pointed already a bit out. Let's see. This is the one domain, the DNA, and this is the other domain. So there's a lot of steps here we know now. Many more than these 20 adapters. There are probably 2,000 adapters involved in this process. But you get then a transformation of your information from one-dimensional in the DNA to globular in your protein. And it can change over time, so it's 4-dimensional. So we see a dimensional transformation from 1 dimension to 4 dimensions. And this is something very, very unexpected. Because you add up and add up and add up information, because a globular system, which changes over time, contains much more information than a linear system that doesn't change over time. So we have different transitions of information, and it increases your information. This is which I will jump over. So DNA itself, you can say, is not a genetic code, but the set of correspondences between sections of nucleotides. This is the coding part, and the specific amino acids makes the genetic code. So this whole set of instructions is, in fact, the genetic code. Coding, decoding. The DNA itself doesn't contain anything. It's just a whole bunch of symbols. There's nothing in it if you don't translate it. We have been talking and discussing lately here in this last couple of days with scientists. We discussed already in apologetics, for instance, fine-tuning the universe, fine-tuning. I tell you, in biology, we find fine-tuning everywhere. All systems are fine-tuned, optimal. One of them, the canonical code. The canonical code is the code as we find them in all organisms. Evolutionists say all organisms have a common ancestor because of the canonical code. The code is the same in all organisms. If you look into a bacterium, the code you find is the same code as in humans. Well, that was a very, very, very strong argument for common ancestry of all organisms. That is, if it would be a random code. The code is not random. I'm not going into the details of the code, but this is the code. How it is connected to the protein amino acids. Here we see a distribution, how it is connected to the amino acids. And you see 6 times of 3 amino acids, 4 times these, 3 times these 2, 2 times these 9, and only 1 time a start codon. And actually can think of billions and billions and billions of different codes, of different distributions. People looked into it, and what did they find? They find the best of all possible codes, optimal for the buffering of mutations. It is anti-evolutionary. The code is constructed in such a way that it's optimal and prevents change. Fine-tuning. It is 1 out of billions who does this. Fine-tuning in biology.
Just for understanding, you mean random change?
Yes, of course, yeah, yes.
Change by purpose.
If we have to believe our evolutionary friend, they say random change, and then you get evolution. Random change is not changing your proteins. So it is optimal for buffering the information in your system. Of course, every now and then something goes wrong, and then you have a disease or so. But 25% of your mutations are not neutral, as they say. So there it goes. Okay, as a molecular biologist, I studied genes a lot. And this is from my book about 20 years ago. This is my log, my lab log. And we studied in those days the CEBP-alpha gene. There was something funny on the CEBP-alpha gene because it is one gene. It doesn't have exons, so you cannot recombine novel proteins from it. And nevertheless, there were more than one proteins that I found in my experiments. There are 2 or 3 proteins. It's not having introns. So what's going on? So I studied very deep, I studied the code. So I could read the code, in fact. You can read the code. You get this whole bunch of letters, A, C, T, G, but we can read it because we understand part of the code. And I discovered several things in this code. For instance, here you have a sort of loop that forms. And a loop, when a loop forms, I can show that here, something like that. You know, it is a regulatory loop because a protein interacts with it. So if you find a loop in a coding sequence, it is extra code added to it, usually. Because this is where proteins bind, preferably. As you can see, I made some notes, but you can also see in the RNA this blank region, unknown property in the 20 years ago. What does it do? Junk DNA? No. We now know regulatory code for microRNAs. This code, encode, encode. What do? They bind here and stabilize your RNA products or destabilize them, depending where they bind. So your products are regulated and controlled on all levels by genetic code. We have to keep in mind that we have different codes in the DNA. I already showed you the genetic code, which is actually has to be translated in protein. This is coded information, protein code, for instance. But we have also what we could call regulatory code or template code, for instance. It is a template because here proteins can bind to, or RNAs can bind to. So this is template code. And it turns out that we have a whole lot of template code in our genome. A whole lot. Much more than code that has to be translated in protein. That will be about 1 or 2% of our whole genome. But we have regulatory code. A whole lot of it. I always say everything in biology is regulated and controlled by codes. Nothing happens just so. We used to think, okay, it happens, but it turned out that everything that happens in a cell is controlled and regulated. If it is not controlled and regulated, you will have diseases. So it has to be regulated. You cannot just express a protein. No, a protein has to be in the cell in a certain amount and in the right location. So everything has to be controlled and regulated. And we discovered a lot of code. We discovered transcription codes. Not one, A lot. We discovered translation codes. If you have a transcript, it has to be translated into protein. More code. Splicing code, for instance, activated codes for translation, RNA editing codes, regulatory codes, histone codes, for instance. I will show briefly what a histone code is. Structural codes. A lot of our genome turns out to be structural codes. What is a structural code? To build the chromosomes, for instance. You only need the chromosomes during a very short time span in your cells, when the cells divide. Nevertheless, you have to code it. You cannot do without, because you cannot make chromosomes and you cannot divide. It is coded information, structural information. Genome stability codes, nucleosome positioning codes. I will explain it to you later. I will look into a few codes that we discovered over the last couple of decades. Code for recombination and code for genetic switches. Transposons. Transposons is the archetype of so-called junk DNA, leftovers of the evolutionary process. Viruses, integrations. No, it's not. These have functions. And I describe actually in my book a lot of the functions that they have nowadays. But I will go into one of the interesting codes that are on top of other codes too. It is so-called nucleosome code. And what does it do? Well, we have to look into the genetics a bit more. Here we see the letters of the DNA, A, T, C, and G. And you know all this beautiful double helix as the DNA. And I think if we think about DNA, we see this thing, this double helix. But it has to be wound if you want to control your chromosomes, if you want to condense it, or you want to spread it out, you have to control it. If you want to have your genes accessible, it has to be spread out. And if you want to close them, it has to be tightened. And this is regulated. But this is about a code. Here you see the DNA. It is tightly wound around these so-called nucleosomes. And how can you wind it? It's not just winding. You need interactions. You need a code for that. And we discovered a few years ago, this code. It is also a code. It is on top of the other codes. If you have a protein code, every turn around these nucleosome, it has code. It goes like this: GC, TA, GC, TA. It can also be TT or AA. It has to be every 12th position, this code. Otherwise, it cannot attach to your beads. So this is another code on top of the code. So in all genes and all DNA, this code must be present, otherwise it cannot bind. So there's another code that I will jump over. Everything in biology is regulated and controlled by codes. I will show you another example, the histone code. It is associated with the DNA. It is not in the, most of it is not in the DNA itself. What does it show? I show the same figure because we also have to go back to the nucleosome. The nucleosomes actually have another code in itself. It's also coded. It's a coded protein, in fact. And what does it see? I can blow it up for you. This is, in fact, what we call the histones, and they form these nucleosomes. And they are built of 4 times 2 proteins. And we call them H2A, H2B, A3, and A4. But it's unimportant at this moment. It's just a small bead. And what is coming out of these beads? A little tail here. And they all have a little tail. And you can modify this tail with small molecules. And this is a code. What does the code say? To be accessible and to be less accessible. The program can be accessed or the program is closed. It is code to regulate your genes, your programs, the right programs in the right cells. This is the epigenetic code or the histone code. And we can modulate it in several ways: methylation, phosphorylation, acetylation, ubiquitinylation, or ribosylation. And you can imagine you have 5 different molecules that you can simply attach here on 20 different positions. Here. So you have a code. Here you have a code, and here you have a code. And it means, the code means, I go open, I go a bit open, I go very open, or I close completely, or I'm open a bit. So it opens up your programs. And it's tightly regulated. And here we start to understand the code on this H3. We studied it a lot. We start now to understand it, how it works. And there's one thing that when this one, the K27 lysine, it's an amino acid in the protein, when it's methylated, the whole program closes. This is what we know now. So if this one is methylated, so this very small molecule is attached to it by enzymes, which is regulated and controlled, the whole program switches off. So this is what we know at this moment. And of course we know a lot of modifications already, but we do not really understand how and what. But it has to do with opening the programs and closing the programs. Because you can imagine we have so many different types of cells that you need to sell different programs that are activated and silenced. This is epigenetics. In a heart cell or in a skin cell, you completely have different programs activated and closed. And this is regulated by the histone code or the epigenetic code. Okay, a lot of codes, as I said. Structural codes. This is also very important and I think very recent. It's not so far. Everything I talk about is the last 2 decades. This is a so-called DNA structure code, and it determines where the chromosomes are found in our cells, in our nucleus. We used to think they're located at random in the nucleus. But if you look at the same type of cells in the same stage of development, you always find a similar pattern. As you can see here in more detail, and you can see all our chromosomes here, and they actually are present in space. And you can predict it, stage and space presence, because it's regulated by code. And what is this? What sort of code is this? This is just sequence code that can interact. So this is not a code that you have to translate. No, it is an interactive code. It controls the 4D pattern of our nuclear DNA. And it involves a lot of repetitive sequences, which we used to call junk. And if we look into these repetitive sequences, which we did not analyze in the past because we were so focused on exons, you see big differences here between chimpanzee and humans. Up to 70% differences. We don't even find them in chimpanzees. So this 3D structure of our chromosomes are very important and very specific to specific organisms. So, if this is information in this letter, which is just a row of symbols, is it also information in the DNA, which is also a sequence of symbols. In our written language, we have 26 symbols, which we call the alphabet. In the DNA, we have 4 symbols, which we call the alphabet of the DNA. A, C, G, and T. So, is there information in the DNA? My answer to this question is yes, because I can read it, I can translate it, I can understand it. I say there is information in it. But what is information? And if you look into information, the first one you will encounter is Shannon, because Shannon was, I think, the #1 person in information theory. He was a US-American mathematician who worked for the Bell Labs, I think in the 50s, 40s, 50s. So just after the discovery, I think, or just before the discovery of the DNA. And he had the question how to transmit signals most efficiently across telephone wires. And then he adapted entropy, because he realized that it had to do with something to do with entropy. The more chaos there is, the more information is there in the system. And the more ordered the information is, the less information is in the system. So it had to do something with entropy. It actually comes up with a very, very similar formula. But I recently watched him. I thought, okay, what does the man say himself about his discovery? And this is what he says in an interview, one of his final interviews he gave. And he says, information or intelligence, as they called it at that times, was a slippery concept. And he wanted to find, or he says, I wanted to find a way to treat information in a physical way, like energy, which you could measure. So you have to look into information without meaning. Meaning is irrelevant. To transmit it, for instance. And then he gives us an example. He says, coin tossing is an example of the simplest form of information to communicate. It can either be heads or it can be tails. So if you know it is heads, it can only be tails. So there is a very low amount here of possibilities, heads or tails. And he realized it's true or false, or 0 and 1. So you have a binary system, and this is what they work with in telecommunication, off on. So he realized this, and so he could come up with his information theory. This is in fact what I said. It is an off-on system. Every message can be written and transmitted as binary code, as electrical signals. Message, you have an endless amount of signals, messages. That's what we call the message source. There's a set of all possible messages that can be sent and received. The message itself is just a string of numbers, symbols, words. And he said, okay, this is then the information content of the message. Why log 2? Because the most simple system has 2 possibilities. So if you say log base 2 is 1. So that's the most simple information system, has 1 bits of information. And this is why he formulated it in this way. So it's a sort of informal, an intuitive formulation. So informally we can say about this information is that the information content of a message is the amount of surprise or uncertainty the receiver has upon receipt of the message itself. And I already spoke about this coin flipping. You have only 2 messages here. It can be either heads or it can be tails. And this is the most simple information system. 1 bits of information. So let's have a look at babies. How much information do they transmit when they start talking? The baby boy says, da da da. How much information is in there? According to Shannon, there's only 1 message. It's not complex. Log 1, 0. This boy doesn't transmit any information because you can predict it completely up front. He says da, 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 da. There's no information in it, according to Shannon. The baby girl says ma pa da. That's more complex. She's sending out more messages. And she's sending out how much? Well, according to Shannon, 1.58 bits. The point is, Shannon information doesn't have meaning. It does not have to have meaning. But da da da, it has. Yeah, it has meaning. Absolutely true. Yeah, so let's look at the DNA. What is the information then in the DNA? And if you look just at the position, one position in your DNA, there can be 4 messages. The message source is 4. So every position in your DNA has 2 bits of information. This is how you define it. If you look just at the individual letters. Every position contains 2 bits of Shannon information. So you could argue that our genome has how much? 6 billion times 2 bits of information. So that's a lot. The thing is, if you look into sequences of information, it's going to be different. It's going to be an add up, and this is also from Shannon. And then we have to calculate it. And this is, I think, an interesting example. How much information is in this word, schoolmaster? Well, you can add it up. You can look up the relative frequencies of all these letters. You know the message sort can only be 26. So you can calculate on it with this Shannon formula. And then you find about 2.02 bits of information in schoolmaster. This is just Shannon information. It does not say anything about meaning, because if we know schoolmaster, we know what we talk about, isn't it? So it has no meaning. Shannon information has no meaning. I will show you that by an example. We can scramble these letters into the classroom. You can do it. It's exactly the same letters. The classroom and schoolmaster are an anagram. How much information is here in Shannon information is in this system? It doesn't change. The letters don't change. The relative frequency do not change. So the information is also not changing. The same amount. Nevertheless, we know it's not the same. Schoolmaster is something completely different, has a completely different meaning than the classroom. So Shannon information has nothing to do with meaning. And this was also how he set it up. He said, I'm going to couple it from meaning. So, important lesson, Shannon information does not have meaning. It doesn't even have meaning. Let's have a look into the DNA. This is a very short sequence of 9 DNA letters. You can also do the same for this DNA, this very small part of DNA. How much information is there? You can calculate it. It's about 1.8. So now I'm going to change the order. How much information? Same letters. Nothing happens to Shannon information. So it is also 1.8. Shannon information doesn't tell us anything about meaning. Let's have a look in biology. If we look at these two sequences, they have a completely different meaning. It is a code. It has Shannon information. You can describe it in Shannon information. It has a very low content of Shannon information. Nevertheless, it has meaning. It has different meaning because if you translate it, you see that different amino acids are being formed by these two same Shannon information molecules. So you can say, okay, we have Shannon information, it is Shannon information. It does not address our biological information. Shannon information does not have to have meaning, and we see that in biology, this type of sequences have meaning. They have meaning, so it is not Shannon information. I can put my whole genome in my pocket on a memory stick. I can never from this Shannon information that I have, I can never make a human being. Never. It's impossible. On the other hand, it's like this. Our genome can be simply downloaded as digital information in binary code. And this is the easiest form. You can simply write down an A is double zero, a C is 0, 1, a G is 1, 0, and then you can end up with a very small description of your DNA. And this is in fact what you do, a bit more. In fact, when you use the ASCII code, the standard American ASCII code for all these symbols, you will see that A, T, C, G, our building blocks, is just a whole row of ones and zeros. So in fact, we are dealing with a quaternary code. Binary code can be transcribed into quaternary code. So our genome is built of quaternary code, coded information, which has a meaning. So basically, we could write everything what we see as zeros and ones, but the other way is also possible. We can also write all information that we gathered and that we stored in our computers. We can also write as DNA in a quaternary code, and we condense it. We condense the codes. And people do all work already on DNA computers, but also on DNA storage systems. Because everything we produced, all information systems that we have on the world would fit in this room when we simply store it as DNA. It would fit here. Well, if you look into storage of electronic information that we now have, it's huge. So we can condense it in DNA. It would fit in this room. So you can imagine that scientists, computer scientists, want to have a DNA computer in storage. You can also freeze it down. It will never degrade. You simply can upload your computer, your whole computer, in bacteria and decode it later. You can keep it for ages, deep frozen. So they look into this, of course, very hard to understand. This is another example. I think you understand it now. The string of letters contain a lot of Shannon information. I'm not sure if people here in the room do see something familiar. I don't know. You just said da, that has a meaning, give, da. Okay, but probably people with a different background, language background, recognize the signal. I just took random letters here. And for me it doesn't, it has of course Shannon information, but no functional information. And this we recognize immediately. This is functional information. So the string of letters has about the same level of Shannon information, but it has function meaning. So this is the difference. This is the difference. Meaning. Okay, we know that as creation scientists, we know this, that there is meaning in the biology, because in the 1990s and even earlier, some people like Werner Gitz in Germany already thought about it, and he came up with books about information in the cell. And he already started to address it as language. And you see Eugene Koenig, he's an evolutionary biologist. He also comes now up with this concept. He says, okay, the information in the genome is not just Shannon information, which merely refers to the mean entropy per position of a nucleotide. No, he says, it is meaning, as in language. So more and more people start to realize that we are dealing with language, with programs, programmed language. Eugene Koenig, he says, how do we find this? How do we find this meaning? How do we extract this meaning? And he says we should compare organisms. Like 4 organisms, and you compare the DNA sequences, because it is code, you can align it, and you see, okay, we find the same sequences in 4 organisms, and he says these are called homologous sequences. He says this is functional. This is the functional information of the DNA sequence. So in fact, he implies that the other part, though this is information, and in fact he implies that this is not information. It has not been conserved, he says, so it's not information. So what does it mean? For me it means you miss most of the information of your genome. You miss all unique information in your genome. So this is a sort of Darwinian presupposed definition of information. All unique sequences that you find and that set us apart from all other animals, for instance, for apes, we have a lot of unique information in our genome. You will not find it by this type of defining. All unique sequences will simply not be regarded information. So it's not a good definition. There's another one. This is called Jack Szostak. He's a bioinformatician, mathematician. And he says the same. He says the information content of biopolymers, so DNA and RNA, is usually thought of in terms of the amount of information required to specify a unique sequence or structure. This is in fact Shannon's information. And he says also this viewpoint derives from the classical Shannon information theory, which does not consider the meaning of a message. So people, they start to realize that they are dealing with programs and with speech and with language. So there are languages, there are programmed languages in our genome. And he says functional information is not a property of any one molecule, but of the assembly of all possible sequences ranked by activity, and more information is required to specify molecules that carry out difficult tasks such as high affinity binding and rapid catalysis. So he's going in the direction of function. He wants also a functional definition of information. So you can summarize it as this: bioinformation is proportional to function. It is related to organized or functional complexity of the biomolecules. Now the next problem comes if we realize that it is functional, that it is proportional to function. How do we find function? How do we find it? Is our whole genome functional, or is it 80% or is it 60%? The most recent data that I have seen is that it goes in the direction of 80%. And this is very, very contented. People do not want to have it 80% because they know if it is 80%, evolution could not have happened. This is the point. But it's fairly contented. Okay, so how do we discover function? How do we measure function? Well, you can do experiments. For instance, you can knock it out. And you probably, if you knock out certain parts of your genome, and you look at the fitness, for instance, of organisms or of functional disturbances or shortcomings of the organism, anomalies, then you can say something about a sequence. The organismal fitness, you measure the fitness. How fit is it? How good does it reproduce, for instance? Does it have observable fitness effects? Well, in the 1990s we discovered we cannot do it like that. Why is that? Because it is the so-called no phenotype knockout. What we did in the 1990s, and I was involved in this type of research, we wanted to know functions of what evolutionists call highly conserved sequences in our genome. Mice have the sequences, but also humans have the sequences, which speaks out for function, because it must have been conserved for almost 80 million years. So it must be very, very, very important information. So what the functional genome biologists did, they knocked it out in mouse. So they simply removed the gene from the mouse, and then they hoped for phenotypes, functional disturbances. Surviving a knockout blow, what does it mean? You can simply do that. And they show up no phenotype, although we expected it. It was one of the biggest surprises in the 1990s. And it showed that selection does not really have a very important role to preserve this information, or these organisms cannot be 100 million years old. This is what we have to choose from. In many cases, there was even a journal who dedicated a whole area of the paper to the no phenotype knockouts. And of course, there's a lot of disbelief because where does this information come from? How is it stabilized over 100 million years without selection? Big questions. They have not been addressed thus far. Of course, you can model it in mathematical models with very, very small selection effects, but you need huge populations. This is the point. So there's a big problem. Now you can say, okay, that's just an artifact of the laboratory. No, 2014, Science reported that all human beings miss 20 genes, randomly you can say, because we have a lot of genes that are inactivated. They do not function. And if you get one from mama and one from papa, you are a knockout, like these mice. So this was published in a few years ago. We are also knockouts. Every person here in this room is a knockout for 20 genes. And we don't even see it. Well, maybe we see it. Maybe it has to do with aging. Maybe that's what it is. But we do not see it. Otherwise, we say everybody is normal here. We are not, we think we are not sick. Nevertheless, yeah, we hope we are not sick. Nevertheless, we miss 20 genes. And not all the same. All persons miss different genes. That's quite something, isn't it? So on average, every individual lacks 20 genes, and we are carriers of at least 100 genes that are missing in the so-called that you inherited only one missing gene from one of your parents. So I summarized this in my book because I found it so astounding that there are genes that you can knock out, and there are actually genes that people think they're conserved for millions and millions of years, and sometimes it is huge sequences in our genome, 2 million letters. You can remove, you find them in mice, you find them in humans, nothing happens. So I think this is a major problem in our thinking, our evolutionary thinking. So I described this very extensively in my book, and what it means, and how you can solve it. I solved it in my book. But then you have to simply completely go away from Darwinian thinking. You need a completely different view of biology. So the information in the genome has to do with meaning. This is what we saw, as in language. And how is a language built up? And I will compare now what we find in our genome to a language. Here, this is from Wernicke. It was published in 2009 in the Journal of Creation, but he also wrote books on this topic very extensively. What is a language? It has 5 levels, he says: statistics, syntax, semantics, pragmatics, apologetics. And we see that also in our genome. We see DNA letters, that is the statistics, that's the statistical level. We see genetic code. The DNA contains words, which is the grammar, in fact. Description, the semantics is meaning, description of the amino acid sequence for proteins, regulatory RNAs, and so forth. This is the semantics, the meaning. Then they have to evoke an action, which is the pragmatics. You have to activate the biological system together, for instance, some metabolic system, or an apoptotic system, or whatever. We have about 400 basic biological systems that you have to activate. So this is the action, the pragmatics. And then it has to have a goal, purpose, result. And the goal is to keep life as life is, to propagate. So DNA is a language, the language of God. There's a book, Francis Collins. I think it's worthwhile reading because he also shows it's a language. I will skip that because I think I'm lacking a bit of time. But yeah. Yeah, okay. Okay, bioinformation, a novel gene concept. I already talked about it a bit. This is how we thought about a gene. It is a coding sequence, the exons, and they are then introns. And the introns have to be split out. But the DNA now is completely different. Since about 10 years, 15 years, this is what a gene is now. There's a lot of regulatory units, and the promoter, as we used to think the gene is driven from the promoter, as it is in bacteria. No, in the higher organisms, as we call them, a gene is diffusely spread through the whole region of your chromosome, and is regulated by 70 to 100 elements that we used to think of as junk DNA. Most genes are driven from so-called transposons, and transposons are now being regarded as enhancers or promoters at a distance. So you have to build up, in fact, a whole platform of sequences to induce your gene expression. So we call them I call them at least. I wrote here an article about this topic a few years ago with a colleague of mine. And we call them protein coding islands in an ocean of regulatory units. This is in fact what the genome is. very diffusely spread information, which is then built. It is built up. In fact, you build up your program in real time. You get a signal. What protein do we need? Okay, that protein. Then we build up a platform where we can find all the things. We build it together. It's like a factory. Cell is really like a factory. You combine and combine, recombine, and then you come up with a protein which is functional in a certain situation. So it is an unbelievable computer. And I already mentioned that. So here, what did we also discover as coded information in our genomes? A whole universe of small regulatory RNA molecules. And here we have the old school. We knew about a few RNAs, but the deeper we look with this deep sequencing, the more we find. And some are present in very low amounts, hundreds to thousands, and they have functions. This is the new RNA universe. I did a lot of work myself in this area, but we are not going to look into it. RNA are part of the background operational programs that command and coordinate activation and deactivation of genes. We have very, very tiny amount of coding regions, which are the tools, but the tools have to be regulated and controlled and expressed at the right time, in the right cells, to do their function. And this has to be completely regulated. By what? By code script. So it's a computer. It's a very, very, very similar to a computer, as you can see. Left-hand side, the human computer to build machines. The right-hand side, the human computer, God's computer, the cell. It gets the signals from the outside and it is replying and responding to make these beautiful creatures. The computer code is a pattern of symbols that carries information to specify machines, and the genetic code is a pattern of molecules that carries information to specify organisms.